In this video, we will discuss about how this CRISPR scan identifies negative regulator of CD8 plus T cells in tumors. So for this, they first prepare a gene knockout libraries. So in order to prepare the libraries, how they did is they first select all the genes that are present in this CD8 naive CD8 plus T cells, and then remove the genes that are necessary for survivals, and then took out the other genes, and their focus is mainly on the regulations of these T cells. So they find out around 899 genes are important, and they target 3 guide RNAs against each gene. This is how they create these knockout libraries. So what they did is they first took the cells and transduced them with their knockout libraries and lentivirus of their knockout libraries, and then screen them with this Thai 1.1 reporter, cultured them in for 7 days in interleukin 7, and thereafter they transfer these knockout libraries, or these transfer these knockout libraries in these mice, and after 13 days they isolate the mice at 2 different places, that is from the tumor or tumor derived lymph node, that is TDLN, and then compare the guide RNAs count after these selective pressures, that what is the input and what are the guidelines that they are able to find out in the TDLN and the tumor case. And they did the similar studies but with the activated T cells also. So in that case they took the same Cas9 positive cells and activate them for 18 hours with anti-CD3 and CD28, and thereafter they screen or they transduce these cells with lentivirus transductions, and then sort them with this Thai 1.1 reporter. And also they do the, and for that they do this culture in interleukin 2, 7 and 15, because to keep the cells activated. And they, after day 12, they transduce these cells into the mice, and after day 19 they sort this Thai 1.1 positive and CD44 positive cells, and from 2 different locations, that is tumor and TDLN, and then compare the guide RNAs with the input and the tumor cases. So first, the panel B is about the TDLN case. In that case, we can see that, and they compare it with respect to the input. So they can see that some of the negative regulators that are shown here around STRUB1, GATA, NCTN, and when you check it, and this is panel B is for the naive T cells, that is in the first row, and in the second row we can see that for the activated T cells they are able to see the similar amount of similar genes that are down regulated for the CD8 plus T cells. It is STUB1, GATA, SPN, and when they compare it for the multiple test results, what they are able to see is that genes that are continuously down regulated is mainly this STUB1, PT, PN2, TNA, TNFA, P3, PD, PDCD1, and from the TDLN also they are able to find out almost all the guide RNAs that they have inputted, all the guide RNAs of the knockout libraries that they have inputted. And when they check the results for the tumors or the guide RNAs that are screened from the tumors, they are able to see similar kind of negative regulators of the CD8 plus T cells, such as TNA, FI, PTPN2, STUB1, CATA3, and this is for the naive tumor cells. And when you check it for the activated tumor cells, and here also when, and they are comparing it with the input, since they have also sequenced the input cells, so they also see the similar kind of down regulators genes for the CD8 plus T cells, that is STUB1, TNA, FIP3, PTPN2. And when they did the screen comparisons of the multiple uh, samples, they are able to find out that this STUB1, PTPN2, TNA, P3, PDC, D1, TRB53, AF3H, all these seems to be looks like a probable candidate for the downregulation of CD8 plus T cells. So this is what they have found out in the preliminary studies. So I hope you understood so far till now that how they come up to some of the possible candidates for the CD8 plus T cells negative regulations. So if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.